Hi, in this video I am going to talk about limitations of biodiversity monitoring programs. Here I have listed 7 different main limitations of biodiversity monitoring: poor estimation baseline, species selection, monitoring non-colonies, measuring trends at atypical locations, reliance on relative measures of abundance, monitoring sample plots within colonies, over-ambitious objectives. I will detailedly explain each and every limitations of monitoring. One thing I want to say, most of these monitoring limitations can also be applied for the limitations for the inventory process. First one is poor estimation baseline year. So in our indicators, what we do, we are taking the first year as an indicator, and based on that we are predicting the trends, whether the population is increasing or population is decreasing. So our all the calculations are based on the baseline year. Okay, so in most of the studies, initial year is taken as the baseline year, but sometimes what happens is like there will be some inaccurate measures in the first time, which will leads to complications or errors in the all subsequent estimate. So inaccurate estimate in baseline year can be due to different reasons. Baseline year is the first year of time series. When we are doing the experiment for the first time, there may be some inadequate sampling, or maybe techniques may not be optimal, things like that. So in the baseline year, there may be sample size is low, as we are doing the experiment for the very first time. We are taking the samples for the very first time, maybe the sample size will be less. So if the sample size is less, the precision will be less, the chances of bias will be high. So if the baseline year is biased, all our subsequent data will be biased. Okay, next we have the sampling variations. Because of the fluctuations in the population, maybe there is a chance of sampling variation as it is the first year. Maybe baseline has a small number of species. When we are starting a survey, most of the people starting the survey with a limited number of species. As the survey is established, we will add more and more species into the group. So that can be happen. And in most of the species, there is a chance of annual fluctuation in the index. Some species may be abundant in one year, in next year it may be reduced. There is a natural variation, natural tendency. If we are looking at any species, we can actually see that there is a natural fluctuation. If the baseline year is an year which have the lowest number of individuals, lower side of the fluctuation, the subsequent estimate will be like not that much representative. The other way is also true. If the baseline area is having higher number of individuals, the all our subsequent calculation will be misleading. So here is one example. This one is from LPI. The first year 1970 is taken as the baseline year. Here we have the baseline. So based compared to baseline in 2006, we have there is a 31 percentage increase in index value at the temperate plant index, but in tropical region there is a 61% decrease. What if we have taken our baseline year as 2000? If the baseline year is 2000, the change will be in a different way. Okay, we are calculating from somewhere here. Okay, so based on the baseline year, the all calculations of indexes will be changed. So if there is an error in baseline year, it will be a huge issue. So how to avoid the changes in baseline year? The first thing is that the first year of time series need not be the baseline year. We can actually change. Okay, because once we start the survey, we can do the surveys for 2-3 years, and then we can calculate as the survey is progressing. Survey will be more established, and there will be more precision. After once we attain the required precision, we can select the baseline year. Or there is a generalized additive model. In this, we will add few years together in the initial thing, and we will average the baseline. So instead of taking depending on a single year, we will take a few years combined to get the baseline data. Or there is a 10-year moving window. We are always comparing the biodiversity change when compared to the data of 10 years before. Okay, what happened in when compared to the population size before 10 year, how our population is performing now. So that's another option. Or we can do the estimating the second derivative of the smooth end index. By using these ways, we can actually avoid errors in baseline. Then species selection. The most of the surveys have they are not representative sampling. The thing is that here you can see this is the actual number of different types of species we are seeing. We have this much amount of proportion of plants, we have birds, other invertebrates, insects, moths, and beetles. So this is a population structure in a particular geographical area. But when we look at the indicators, the same area has in the real population, birds constitute only this much. But in our indicators, birds are constituting almost half of it. Same goes for the moths, terrestrial mammals, and the butterflies. Some of the things, the plants are not at all represented in the indicators. So there is a difference in the actual composition and our indicators, which is a huge issue because only few species are represented in the indicators. So based on the indicators, we are making policy decisions and everything. But if the indicators are not the real indicators of the biodiversity change, that will be issue. That's a limitation of biodiversity monitoring. So the indicators we are using for biodiversity measurement should really represent the population structure. Okay, so there are different ways to avoid this thing. First one is weight the index based on the composition of species. Weightage will be given to all the indicators to get it more representative. Then, or we can go for occurrence data rather than abundance data. So if you are going for occurrence data, the frequency of occurrence will be measured rather than the number of species. Okay, the occurrence data will be more representative of species. Then third limitation is monitoring non-colonies. Some of the animals or birds are living in colonies, just like bats. They are living in huge colonies. So there are chance for changes in this monitoring practices. The mainly, if you this is a bat colony, so when we are going for monitoring the changes in bat population, sometimes what happens is like we will be monitoring 2-3 colonies, maybe based on the sampling size, we'll be monitoring uh, some number of colonies, maybe 10 or 20 number of colonies, but we are studying only that colonies. What happens is like there are chances, random chances like that these colonies can be abandoned by all the species. It will be vacant. Nobody will be coming to here. The total abandonment of the colonies. At the same time, these bats may go to other places and establishing a new colonies. But if you are focusing your studies on a limited number of colonies, the abandonment of these colonies will be recorded, but they may be creating their colonies elsewhere, maybe some remote locations, unidentified locations. Uh, in those cases, the loss of colonies will be recorded, but the establishment of new colonies may not be recorded. So, in your estimate, we will be seeing a decline in population, but actually there is no decline in population. It is just the misrepresentation of data. So that is a problem with the monitoring of non-colonies. Monitoring sites are usually selected based on non-colonies only. So we are only following the non-colonies. Once they are abandon the colonies, we will record those things. But establishment of new colonies in a remote area or unidentified area may be missed from your calculations. So that's say an issue for organisms which is living in colonies. It can be overcome by representative site sampling rather than sampling the colonies. The sites should be sampled. The whole area should be sampled based on random sampling methods. Next, measuring trends at atypical locations. When we are like bird survey or butterfly survey, what happens is like we will only survey the region we know about. We know some areas the birds will come continuously, or there will be chance of getting seeing birds, or there may be places where the butterflies are plenty. So the sampling will be mostly happen on those areas. Some other unidentified areas will be always missed out. So that's a problem of measuring trends at atypical locations. This, if you are sampling only a few selected sites, there is a chance that your data is not representative of the whole scenario. So some regions are overrepresented because it may be easy to go there, or people know about that region. If you know some butterfly gardens or places where butterflies are usually coming, or birds, areas where birds are abundant, so more sampling will be occur only in those places. Other places are totally missing. Maybe there are some areas where birds are abundant, but no sampling is taking place. So that's an issue. Uh, Especially when we are using citizen scientists, people will just go by their knowledge of places. So in order to overcome that, we can use different strategies like diversity weight index, or stratified random sampling, or model based approach. By using these three approaches, we can actually overcome the limitations in sampling at atypical locations. And next, reliance on relative measure of abundance. Most of the calculations of indexes, we are actually counting our like representing our data based on a previous year, based on relative to the previous year or baseline year, how much population is grown or decreased. That's the main way of representing. But there is always a chance of change in detectability. Detectability of an organism can change due to many reasons, maybe due to habitat succession, or maybe we are using better techniques. Sometimes the number of organisms will be decreasing, but over the time we have more advanced technique for sampling. So maybe the previous time sampling, the counting the number of tigers is based on their footprints, the characteristics of the footprints. But we have the camera traps, high resolution photography, or imaging techniques using drones and everything, or acoustic methods. If you are using those methods, the detectability will increase. The chance of detecting an animal will be increasing. So even if the population is decreasing, as the sensitivity of your method is high, you will capture all the information. In previous time, maybe the forest having 100 tigers, but your method is not that good, so you are only able to document 50 of them. Half of them are undocumented. But as the population, as the technology advances, even if the forest is having 70 tigers, you are able to detect all of them. So in data will become misrepresented, or phenological changes and behavioral changes. The animal may change the behavior, maybe the seasonal variations or the time of day where when it is active, things like that can change. That can be misrepresented in your data. If you are taking samples at 5 p.m. every day, maybe if there is a behavioral change happen to the organism, it will become less active during that time. Maybe it will be active later on. Your data will be misrepresented. Other thing is that some of the surveys has long run surveys for the 50 years, 40 years, things like that. So they are using same peoples for the survey. So as the people get more age, their ability to detect will be reduced, especially for bird watching and mosquito butterfly watching. So observer age is also affecting the detectability. Other thing is our observer learning. Once we are surveying for a very long time, the observers will know how to find this organism more efficiently. So the data will change. It's more affected by long-term time frame studies rather than the short time frame studies. So these represented the different factors which is affecting the detectability of the organism. It can be affected by species characteristics, spatial variation because of the habitat densities, suitability and disturbances, or temporal variations during the survey, like seasonal variations, phenological changes, weather, time of day, and survey characteristics like detection methods, survey effort, observer skills, time of survey. Everything can affect the detectability. So these all things should be taken into consideration when we are making the index data. So for that, to avoid these limitations, we can actually estimating the detectability. We can integrate the detectability factor into the calculation of the index, or modeling can be used to compensate the changes in detectability. Then we have monitoring sample plots within a colonies. Some of the organisms are lives in huge colonies like this. It's a bird breeding colony for penguins, I guess. So in these colonies, this more or less impossible to count each and every individuals in a population. So what we do, we will take random samples from random locations. So if your random location is here, you see here the number of birds are less. But if your random location is here, the number of individuals is high. So it's a very difficult situation because the counting the number is of this huge populations is difficult. So what we do, we measure the density of population rather than the overall size of the population. But the thing is that even if the population is shrinking, the density may remain the same because it's a characteristic of an organism to be in such a densely breeding colonies or things like that. So in this thing, if you are measuring the density of the population, it may be misleading because even the size is less, it will be maintaining the same density. The counting the random plots is also difficult because of the some plots may not have any organism, but some other plots have large number of organism. Because especially areas like this, the areas which is steep and stony may not have high number of birds, but areas like this may have more number of birds. So this can be a misleading thing. Defining plots on irregular grounds is difficult because these areas may not have that much number of individuals, and other cases density may be staying constant even the population changes, sizes change. So what we can do is maintaining sampling rates across the colony. So instead of limiting the sampling locations within the colonies, we will sample the whole area so that we will get the data about the sampling population density as well as the population coverage. So if the population size is decreasing, there may not be a change in density but a change in coverage. Finally, final limitation of monitoring studies is over ambitious objectives. When we are studying monitoring a population in a small geographical area just like our college campus, we can have a detailed study in very high time frame for every organism. Because the area is less, we need to worry only about few organisms. But consider you are doing the same study for the whole state, the things will change very dramatically. We cannot have a detailed study, we should have a randomized sampling. So most of the large studies will start up with like unachievable objectives, so that will mess up with this study. Okay, so to monitor regional or national biodiversity trend, regional schemes does not need to be capable because the schemes we are using for a small geographical area may not be applicable for the large geographical area. In a large sample of representative site is required. If you are looking for a large geographical area, randomized sampling from different locations using statistical techniques is required, and the field method should be very simple, so that we should get only the essential data. Okay, if you are going to study every biodiversity details in whole area of a huge national boundary or in a like in a state or in a nation, it will become impossible. That's a over ambitious objective. So the data collected from a huge area should be sufficient and simple. Okay, we can also go for the power analysis to overcome this difficulty. So I think that's the end of the fifth unit. Thank you so much for listening.